There we go. And once again, I'd like to welcome everyone to the Los Angeles Birders webinar for tonight. If you're a member of Los Angeles Birders, thank you very much. We really appreciate it. And if you're not a member, please consider becoming a member. Lab membership brings along with it that wonderful satisfaction of supporting a wonderful organization like Lab, as well as getting first dibs on field trips and educational programming, and it lets you interact with a Q&A and any kind of polls that may be going on during our webinars. So please support Los Angeles Birders. And with that, I would like to introduce Kimball Garrett, who will be introducing our speaker for tonight. Kimball.
Okay, thank you, Ron. So tonight we're going to hear about an institution that's very near and dear to me, and the Natural History Museum of Los Angeles County, that is. So I'm very pleased to introduce Dr. Alison Shultz as tonight's speaker. She's been the curator of ornithology, now the associate curator, since 2018. So 6.5 years has gone by very quickly. Allison's a native to Southern California, and she developed a passion for birds when she was an undergraduate at UC Berkeley. And then she later earned her master's at San Diego State University, where she studied the evolution of plumage coloration. Her study organisms were primarily tanagers there. She worked under Dr. Kevin Burns, who just gave a wonderful keynote at the Western Field Ornithologist meeting down in San Diego this past week. So some neat stuff they're doing there. And then she received her PhD from Harvard University, where she researched evolution of house finches, proving that even house finches are worthy of study and interesting. I recall being chastised on the first field trip I ever went on as a kid for LA Audubon when there was a house finch that popped up and somebody said, "What's that?" And I said, "It's just a house finch." And I was admonished, you never say just a house finch. And Alison has helped prove why that's true. Anyway, her research focuses on two main topics: how birds are responding to anthropogenic changes, and also the evolution of feathers. So she explores how traits like plumage evolve under selective pressures and how they signal health and influence behavior. Her research ranges from studying physical traits and the genetic regions behind them to tracking evolutionary changes over time. And in much of her research, Dr. Shultz utilizes museum ornithology collections. She also encourages community science to help monitor bird changes around Los Angeles. So tonight, Alison is going to take us behind the scenes and talk about some of the exciting bird research projects that are going on at the Natural History Museum, with a special focus on feathers, their structure, their function, and the role they play in our studies of birds. She'll also share a recent project that she co-led on understanding the current way that we name birds, specifically focusing on the general categories that are used in the descriptive portions of English bird names. So please join me in welcoming Dr. Alison Shultz.
Thank you so much, Kimball, for that wonderful intro. Oops, not raise my hand, want to share my screen. Yeah, I was looking up when was the last time I gave a talk at Lab, and I think it was 2001. So it's hard to believe it's actually been that long, but apparently it has. So a lot's been happening in my research world since then. And, you know, if you were to go, one of my favorite things to do is just been to go into the collection and see kind of the amazing diversity of birds and especially their colors that exist and really get just inspired by the birds themselves. And the reason my title was a little bit kind of nebulous this time is because I just have so many cool research projects going on. I couldn't decide what I wanted to talk about until just a few days ago. But I decided to tell you on kind of a new aspect of the research direction that I've been going. And that brings us to not just bird colors, but kind of thinking about how light is interacting with bird feathers beyond just what we see with our eyes, and beyond just what bird sees with our eyes. Because last time I talked a lot about bird vision and how birds see ultraviolet colors and how we need to, quantify color from the bird's perspective. Well, we have to remember that light is just one part of the electromagnetic spectrum. And in fact, visible light is just between about 300 nanometers and 700 nanometers, but there are many other wavelengths of light that might interact with feathers just like visible light does. And in particular, one that's really important is infrared light. And so infrared light ranges from about 300 nanometers to 250, to about 16 micrometers, depending on what kind of infrared light we're talking about. And why do we care about infrared light? Well, infrared light has to do with how birds are going to be refracting heat or dissipating heat, which is really important for their ability to stay warm or stay cool or generally do thermoregulation. And so there are really kind of two portions of the infrared part of the spectrum that we care about. The first is what we call UV vis or near infrared light. So that's from about 300 nanometers to about 2500 nanometers. And so those are wavelengths of light that can be either absorbed by feathers or reflected by feathers. Or it could pass through feathers into the body and really heat up birds. Or, if birds are trying to stay warm, they might either want to reflect or to absorb, kind of depending, are they trying to be cool or are they trying to be warm? Now, but there's another aspect of infrared light, which is what we call the mid-infrared, which ranges from about 8 to 16 micrometers, so much longer wavelengths of light. And that's essentially, these are wavelengths of light that are emitted. So this is how, when you see a thermal camera, really they're recording this part of the electromagnetic spectrum. And that's how you essentially send heat off into the environment. So, the space around you is generally going to be cooler than your body temperature. And even if the space around you isn't cooler, space is cooler. So if you are standing in front of the open sky, then your heat, just because of physics, is going to be emanating into space. And so really, when we're thinking about how different types of light is interacting with feathers, we can't pay attention just to what is visible, but we have to pay attention to what's invisible as well, because it can be really important. Now, you might wonder, if we look at, there's a large variation in color in birds. Now I'm just focusing on what we call coloration formed by melanin pigments, or melanin coloration. And birds can range from being really light colored to really dark colored. You might have birds that have different ecologies. They might live in different places, birds that live in different temperature regimes. And we even can have extreme variation within a species, like we see these song sparrows on the right, are all different subspecies of song sparrows, and they're known to have their plumage varying depending on where we live. I'm not the first person to ask this question in terms of how plumage varies with the environment. Glosier's rule exists and states that darker or more melanistic specimens tend to be in warmer and wetter climates, whereas lighter color individuals tend to be in drier and colder climates. Now, a lot of that research has actually focused on is the plumage dark or not from a visual perspective, but really hasn't been studied from an infrared perspective. And if we're talking temperature, then we need to be studying the wavelengths of light that are important for temperature. So does Glosier's rule still apply in the infrared? We don't know. So collaborators and I sought to ask two questions, which is, first, we wanted to ask, across species, do different species of bird with different life histories show variation in the UV NIR absorption or MIR admittance, so the near infrared or the mid infrared? And second, does variation in the UV near infrared absorption or mid infrared emittance vary across populations of birds conforming to Glosier's rule? So do we see variation across species or within a species? To answer this question, a team of scientists, both at the Natural History Museum at CSU Dominguez Hills, Dr. Terry McGlynn and Dr. Megan Barrett, as well as engineering collaborators at UCLA, Dr. Laurent Pilon and graduate student Thomas Lee, we chose 5 species of birds that had different ecologies to investigate whether these populations are variable in the UV NIR and the mid NIR. We chose 1 nocturnal species, the great horned owl, 2 open habitat species, the common raven and the northern bobwhite, and then 2 closed habitat species, the Steller's jay and the song sparrow. Now, there was a reason why we chose these species, because in addition to kind of being variable in their own habitats, 4 of these species are known to conform to Gloger's rule. So they have lighter individuals in drier, warmer climates and darker individuals in kind of more humid, drier, colder climates, darker individuals in more humid climates. The hot and cold thing can be a little bit tricky to parse apart, but so these 4 species were known to have that kind of population variation. And we also chose common raven as kind of our control, because common ravens don't have any sort of known variation in how black they are across their range. Now you might wonder, have people looked at birds in the near infrared or the mid infrared before? And yes, people have started to look at birds in the near infrared, and we know that there is some variation in the near infrared. But actually, to the best of our knowledge, no one has ever studied birds in the mid infrared. And there's a reason for that, which is that the equipment to do so is very expensive. And so you really need to find someone in the engineering department who has this equipment, who's might be interested in collaborating with you on this question. Here's a picture of kind of your typical spectrophotometer that we use, and that you can get a little bit more expensive, but still accessible near infrared spectrophotometers. And this is a picture of the what we call a nitrogen purge Fourier transform infrared spectrophotometer. So a very, very fancy spectrophotometer for measuring the mid-infrared reflectance. And that's one reason why we only kind of started off with 5 species, because these measurements take a really long time to do. So we wanted to ask, is this worth pursuing further, or, is there just nothing exciting happening in the mid-infrared? Well, these are what our results look like. Each line is how much light is being reflected at a certain wavelength of light, and this is for each species. But these can be hard to read. So for our purposes, we calculated 2 things from that. One was the UV near infrared percent absorption. So how much of the UV light and UV near infrared light is being absorbed by plumage, assuming kind of how much energy comes from the sun. And the second was, how much light might be emitted, and how might that vary depending on your feathers. And so this is kind of assuming you had like a perfect black body that's emitting light, knowing how much variation might be possible with the feathers that we measured. What's the percent that could be emitted of that kind of optimal value? So what did we find? Well, we found that common ravens, they have actually the most UV NIR absorption. So this kind of tracks what we thought. Those black feathers just absorb more heat than anybody else. And great horned owls had the least. That was interesting. And, maybe if you're a nocturnal bird, you have just different ways of doing thermoregulation. And because they're not active when there's a lot of radiation coming from the sun. So it was interesting. We had those 2 species, and then kind of our 3 in-between species, the jay, the bobwhite, and the sparrow, didn't have a lot of variation amongst them, although there was some. But what about the mid-infrared? That was the big question. Well, we actually found that there was variation in the mid-infrared emittance compared to other species. So bobwhites had more mid-infrared emittance than everybody else, which was pretty exciting. Everybody else had about the same amount. And so what's really interesting is when you look at these 2 together, our 2 really open habitat species, ravens and bobwhites, have very different profiles of how they might be dealing with heat. So they might be emitting more heat in the mid-infrared. But, ravens might be dealing with heat by absorbing more in their feathers. And that's a little bit counterintuitive, but what they do is they actually absorb it in the feathers, and then the wind disperses the heat before it can go to their bodies. So those black feathers are almost providing protection so that those infrared waves don't reach their actual body. So pretty cool. What do we find? That the most variation across species occurs in the UV near infrared absorption, but there is some variation in the mid-infrared emittance. So there's something to be worth doing there. But now what about within populations? What do we see? Well, if we come back to our 5 species that we studied, again, the 4 on the top conform to Gloger's rule, the 1 on the bottom doesn't. Do we see any variation in the near infrared and mid infrared? Well, we actually find that There's variation in the near infrared in 3 of these species, but interestingly, not all of them are the Glosier's rule ones. And in fact, the common raven was one of the species that we saw some variation. If we kind of dig into these data, in our bobwhites, the Iowa population, so the northernmost population, had the most near infrared absorption. And in the song sparrows, the desert population from California had the least amount of absorption. So this is interesting because even though these two follow Glosier's rule from kind of a visual perspective, they're actually doing the opposite patterns from each other in terms of would we expect that birds that are living in hotter environment to be absorbing or not heat. So not necessarily following the pattern that we would expect. And in the ravens, actually the two that the Sinaloa and the California populations were the same subspecies, and those weren't different, but when we grouped them by subspecies, we saw very different patterns. And that suggests that it's not just the kind of overall color. So these feathers the colors are produced by melanin pigments, but there might be some interesting structure going on with the feathers that we should explore further to understand, why are we seeing these patterns? Why does this one population absorb more light than the other population? What about the mid-infrared? Was there any variation within different populations? And again, the bobwhites were the ones that had some variation. So the Iowa population had the greatest percentage of emittance. The Mexican population had the least. And what's interesting is when we kind of combine that with the near infrared absorption, we see different patterns. So that suggests that maybe birds are trading off different ways of absorbing or emitting their heat amongst these populations. It's also worth noting, while kind of bobwhites I think of as open habitat species, these Mexican populations that we chose, it turned out are actually more closed habitat species. So habitat itself might be really important for driving these patterns of how birds are staying warm or how birds are staying cool in the mid-infrared. So when we come back to our question, do we see variation in the UV, NIR, or MIR emittance across populations? We do see some variation in the UV, NIR absorption, especially. Other species show variation in the MIR emittance, and Glosier's rule does not appear to hold true in the infrared. And so, kind of what are the big takeaways from this? Well, there's a lot more we need to learn. Again, we are only looking at 5 species and only 3 populations within each species. And so, in the future, we're really excited to extend this much more broadly so we can see, are there any rules in the infrared? Do we see kind of common patterns in birds that live in different habitats or birds that live in different parts of the world? And, do we try and understand, how does light evolve across birds, not just from a visual perspective, but a thermoregulatory perspective? So a lot more to come on this in the future, but we're just giving a little bit of inkling that there's something interesting there to be studied. Okay, so I just talked to you about a lot of patterns of kind of how light is emitted, but what we really want to know next is what's causing that variation in terms of how birds are absorbing or reflecting kind of these different wavelengths of light in the infrared. And to understand that, we need to come back to feathers. And so feathers, if you might remember, are made up by a couple of different portions. What's important here is we've got the plumaceous part, which is the downy part of the feather. We have the pennaceous part, which is the colorful part of the feather. And typically we think of the plumaceous part as being responsible for kind of thermoregulation. Think about your down coat. Down coats are really warm because they have a lot of these really downy feathers inside of them that are going to be just absorbing your body heat and kind of creating this layer of warmth around them. Now, we also might care about the shapes of the barbs and the barbules. There might be variation, in terms of the lengths of these barbs, in terms of the lengths of the barbules. And so maybe this is important when thinking about how birds are retaining heat and the colors that they are. Now, collaborators have studied this before. And so Sahas Barvi in 2001 had a paper that he looked at a bunch of birds across the Himalayas and found that the proportion of down is related to species elevation as well as body size. So the higher elevation you live in, the greater the proportion of down, probably because you need to stay warm in those really high elevation places. And so your feathers are going to evolve. And also kind of the bigger you are, the smaller you are, the longer your feathers are relative to your body size. So what if we think about, and that's one thing, so we can think about kind of the proportions, but something that Sahas noticed and something that I had noticed when looking at many museum specimens is that down doesn't just vary in terms of the proportion that exists within a feather, but actually that can vary in its color as well. You can have birds like the Gouldian finch that have really, really light colored down, and you can have birds that have really, really dark colored down. And so what might be driving kind of that color of that down, and how does it relate to thermoregulation and the ability of birds to stay warm? So Sahas and Dr. Carla Dove and I started a collaboration to investigate this in those same birds that live in the Himalayas. We had kind of three questions that we thought that maybe we know that melanin strengthens feathers. And so is having darker down really important for structural integrity? So maybe, if you have longer barbs that you might want to have kind of more, make them be, less compacted so that they're more structural, they're holding more heat in, they're enhancing that kind of heat layer. You might expect maybe darker feathers feathers that are darker because they have longer barbs that need to provide that structural integrity. Another possibility, we know that melanin absorbs solar radiation. Other studies have shown that birds that have lighter feathers actually might have dark skin to protect them from UV radiation from the sun. So maybe we see a trade-off where if you have, a darker outer layer of feathers, maybe you have a lighter color downy part of the feathers, or the opposite. Maybe if you have a lighter color pennaceous part of the feathers that's going to let more solar radiation through, you might need to have darker down so that can protect you from that solar radiation. So that's another possibility. Finally, we know that melanin absorbs heat. So maybe it's really important for thermoregulation. So maybe if you need to stay warmer, you need to have darker down because you need to absorb more of that heat. So that's another possibility as well. So again, to do this, we measured 149 Himalayan species, and we measured both the brightness and the brightness of the pennaceous part of the feather. So again, that's the color part of the feather. We measured the brightness or the darkness of the down part of the feather. And we measured kind of how long the feather was. We still had those measurements of how the proportion of down in the feather. We also measured the length of these downy part of the feathers so that we could try and look at all of these hypotheses that I just mentioned and see which one holds true. So with all these measurements, we decided we had kind of 5 things that we were testing. We did this both in the back, because you might imagine that the back is maybe more exposed to solar radiation, so there might be different sorts of selective pressures, compared to the breast, which really might be more important for signaling or for things like incubation. So we might expect different things to happen on different parts of the body. So the first thing we wanted to look at, do we see evidence of a genetic correlation between the down darkness, that DD means down darkness on the back and the breast, or the brightness overall on the back and the breast? So we're just asking, if you have a certain color on one part of the body, are you more likely to have that same color on a different part of the body? This little squiggly line here, this little tilde, just means correlated with. Don't worry about it too much. I just wanted to show you some of these things, these kind of equations that we were actually looking at. Then we looked at the pennaceous, or the colorful part of the feather, and we asked if brightness, either of the back or of the breast, was related to elevation, which we're using as a proxy for temperature. We then thought about, is it thermoregulation of the down? So do we see the darkness of the down related to body size, to maximum elevation, to the overall feather length, which are all things that had been found were really important for the proportion of down in the previous study. We asked if structural integrity is important. So do we see darker colored down feathers being related to either longer parts of the downy part of the feather, so more down, or do we see that related to the length of that downy barb? So are the barbs need to be stronger because they're longer? And 5, that solar trade-off question. So do we see a trade-off between how dark the pennaceous part of the feather and how dark the downy part of the feather is? So we tested all of these correlations in the same time, kind of taking into account the relatedness of all these birds to each other. I'm not going to go into the statistics, and I'm just going to show you that all of these things are correlated in different ways. Each of these lines represents a correlation, and so you can see that many different things are related to many other things. Up here, this is the visible light results. This is infrared light results, and you can see that the results are fairly consistent both across visible and infrared. And don't worry too much about what these lines are. But when we look at these are regression coefficients, so anything that's above this has like a line above or below this dotted line, we consider significant. What's really key is that we have a bunch of down thermoregulation hypotheses that are significantly correlated. You can see that there are some solar trade-off hypotheses that are significantly correlated, and one of the genetic correlations. Now let's kind of think about this in the context of our hypotheses that we were just talking about. I've crossed off all of the ones that were not correlated, and you can see that, interestingly, the down darkness is highly correlated in different parts of the body, which suggests that there might be kind of genetic constraints in what color your down can be in one part of your body versus the other part of your body, unlike the overall brightness, which maybe has more evolvability because maybe there are different types of selective pressures. So, is it because of just selection for down darkness is the same, or is it because they're genetically correlated? That'll be something really exciting to look at in the future. You can see our pennaceous thermoregulation color doesn't hold true at all, but our down thermoregulation does for most of the correlations that we studied. Structural integrity, no evidence for that. So no evidence that down darkness is related to the length of the barbs or the proportion of the down. And our solar trade-off does also hold true. So the darkness of the down is correlated with the brightness both in the breast and back. So that's intriguing as well. So, overall, did we conclude? Well, that down color is correlated across the body, while overall plumage darkness is not. And that down color likely plays a role in thermoregulation, and that might also be protective against solar radiation. So this, kind of coming back with our other sort of infrared and mid-infrared results, I'm really excited to pursue all of these questions in the future, because we really need to better understand patterns of infrared reflectance, both across and within species. Because, we think about as the climate changes, do these birds have the ability to change along with it? Can they adapt to a changing climate? And would feathers be part of that? Or are feathers not evolvable enough to actually adapt quickly enough, so birds are going to either need to do something else, adapt a different way, say through metabolism or their ability to, keep warm by other means or to be cool by other means? Or do they have the ability to evolve, longer feathers or darker feathers or feathers with more greater pennaceous part of the down? Who knows? So a lot of exciting ways that we can think about that, and also really important things to study. I also want to just make a plug, it's really fun when you get to talk with people outside of your sphere of influence, and that's not just true in regular life, but it's also true in the science world. So I was really excited to collaborate with some engineers who really made some interesting work possible. And I just wanted to acknowledge kind of my collaborators for this part of the talk. And before I get into the next part of my talk, as well as the museums who let us use their collections to do all these measurements. And that next part of the talk is kind of a different sort of project that my collaborator, Aaron Morrison, and I just kind of finished analyzing and putting together. And this is what we called the Bird Name-a-thon, which was categorizing bird names using crowdsourcing. So this part of the talk, I'm not getting into changing bird names, but if names are going to be changed or not, we should really understand how birds are named. And what do I mean by that? Well, Erin and I, we both study bird color, and we were sitting around talking, we were thinking about, how many birds do you think are named after traits that are only in males and not in females? Erin and I both study female coloration explicitly. And so we were just curious about this question. A scarlet tanager would be an example of this, where you would see a scarlet tanager, you would see a female, you wouldn't necessarily associate that bird with being scarlet because scarlet is a color of red. And so we thought, this is an interesting question. We should just spend an afternoon, flipping through the birds of the world, looking at each species and seeing how many are named that way. It ended up being a much more involved project than that, but one that actually ended up having some very cool results. So kind of we started thinking about what do the common names of birds communicate about species of birds? Here are a bunch of birds in the genus Icterus, 16 of the 32 species. And what do I mean for the rest of the talk, when we talk about the, kind of the part of the bird name that we're categorizing, what do I mean? Well, we are thinking about the descriptors in species common names. So in the Venezuelan troupial, we would categorize that first word, which is Venezuelan, as being assigned to a geographic location category, whereas the orange-backed troupial, we would take that first part of that descriptive, the orange-backed, as being a physical trait of that bird. So if we come back to our birds in the genus Icterus, we can see that a number of them are named after some sort of physical trait. A number of them are named after some other aspect of their biology. Many of them are the geographic location where they come from. And then a number of them are named after something else, generally kind of human culture inspired versus necessarily something that's tied to the bird. So we ended up developing these different categories that we classified bird. We thought, could kind of would encompass all of the different ways that you could classify a bird. And we categorized them as, things related to physical trait. And we ended up dividing that into physical traits that were found in both sexes, physical traits that were found only in males, or physical traits that were found only in females. As well as size of the bird. Is it something about the size? Then in terms of bird natural history, we had behavior. So is this a descriptor that's related to some sort of behavioral trait like song? Geographic location, so this is the specific name of a historic or current location on a map. And natural history, so this might be related to habitat or diet. In our kind of last category, we had a person's name. Is a species named after a person? Is this species named after a local language or the phonetic English pronunciation of a local indigenous term? Now, I will asterisk there, which is that we don't know necessarily what those terms are translated as. So they might actually be related to behavioral or physical traits, but we don't know for this purpose. So we categorize them in their own category. Or miscellaneous. So the descriptor can't be placed in other categories. And I just wanted to give a shout out for Guinevere Pandolfi, who helped us run this project and who helped us develop our methods. There are things that we as ornithologists might commonly know and might not realize that you're kind of, everyday person that doesn't think about birds all the time might not know. And so we ended up recruiting many different individuals to help us with this. We split all birds up into 2 duplicate data sets, and we recruited individuals who were both professional ornithologists, individuals who were amateurs or birders, and individuals who were undergraduate students at New York University, where Erin was teaching. Interestingly, you can see that even though there were the fewest, there weren't that many professionals, the professionals categorized the most number of birds, whereas students generally, we said that anybody who categorized at least 100 birds would get authorship on the paper. And so students generally did that many and then they would stop. And there was a lot of variation in terms of this is the number of species categorizations per participant. So we had some professionals who went real above and beyond and categorized many thousands of bird species, as well as, people who ranged all over the board. So 15 of the 56 students did this on their own time, and all 7 of our amateurs were eligible for authorship on our paper, which we thought was very cool. So what did this data collection look like? Well, we had a Google Sheet that participants could look at. So each participant categorized 10 species at a time, and you had to do them in order, so you couldn't go through and just pick out your favorite groups of birds. We categorized the unique descriptor of English common names. We used the Birds of the World database as the first resource. Can we figure it out just by looking at Birds of the World? And then if we couldn't, then we, said, then use the internet, but then record where you found your information. And we said, if you spend more than 10 minutes on a species, just move on to the next one. Some were very difficult to find. There were a few that we did time out, and we just a very, very small handful that we couldn't place in any category because of that. There were some unanticipated challenges with terminology. You might imagine there might be some kind of terminology that's not in fashion anymore that people might not know. But we were surprised that even some students didn't know what the word scarlet meant. And so that kind of goes to show, if we're thinking about communicating about birds, kind of the language that we use might be different to different generations. And so Erin had a very kind of conversation with one of her students where they were talking past each other. She was trying to help him categorize the scarlet tanager, and he just couldn't understand. And finally, Erin realized that he couldn't understand because he didn't know what the word scarlet meant. So I guess students aren't reading their book Scarlet Letter in their class anymore. But that was kind of just an interesting aside that we noticed. I'll also mention that in addition to this database that we're producing, and I'll just mention too that this database is going to be freely available to use by anybody who wants to dig into it and find bird names and their history. But we also developed a glossary of terms as well as a gazetteer of places that, birds have in their names. So the 42 students reflected and were very excited that they were participating in research. They thought it was very interesting. They learned a lot about birds, so that was very worthwhile. But what did we find? What were our results? Well, first of all, even though, we had two different independent people categorizing each bird name twice, 906 species, about 8%, did not match between the two data sets. And it's worth noting that the students did not have a significantly higher individual mismatch rate, so the number of their categorizations that a student made versus a professional. And then 9 species, as I mentioned, had the time limit for both. So we said, okay, let's dig into that. What categories were the most confusing? So by far, the most mismatches were for both sexes physical trait versus a male-only physical trait. So some people said both sexes had it. Other people said males had it. Kind of the runners up there were geographic location versus life history. At the time, we had called it life history. So we did some refining of our kind of explicit directions, and then had each of our 906 species that was not matching categorized independently again by 3 other different people. And these were all professionals. So 10 professional ornithologists independently recategorized these 915 mismatches in triplicate. And then 554 of the 915 were matching by all 3 people. 324 had 2 of the 3 that had matching categories, and 37 had different categorizations by all 3 of the professional ornithologists who categorized them. So Erin and I went through and kind of made the final decisions for all of these based on, for the 2 out of 3, we went with majority rule for the most part. And then based on kind of the justification that each of our participants provided, made a final call for each of those 37 species. So then we had, almost 11,000 birds categorized. So what did we find after all of this hard work of categorization? Well, it was interesting that almost half of birds are categorized by physical traits in both sexes. Almost 1,000 species are categorized after a physical trait found only in males, and only 20 birds are categorized after a physical trait found only in females. Kind of after physical traits, by far the most birds were then categorized by geographic location, and then by eponym, so named after a person. So 900 and something, I'll go to my table next, were named after a person. We can look at these specific numbers here. It's interesting, just the number of unique terms in each category. So there were, 1,530 unique terms that were in this both sexes physical trait category. And the 31% were only used one time, so in one species, as opposed to something like behavior. 74% of the terms were unique or only used one time. And that's probably because most of them were named after a song, and that song's going to only occur in one species. Size-related words were by far the most repeated in terms of their categorization. It was really fun to kind of dig into what were the most common terms used across birds. You'll see black was the most common. And white-throated was another really I actually think white-throated might have beaten out black, but they were kind of neck and neck there. Common was the next most common. African, Western. So it was fun to kind of see what terms are most often used. We then wanted to dig into a little bit, do we see similar results in birds that are maybe related to each other? So maybe birds in certain families. We first took kind of approach across all birds, and we found that birds that are more evolutionarily related to each other are more likely to have names in the same category. So if you are in the same family, you're more likely to have names that are either in a physical trait or in named after some aspect of their biology. It was really fun to kind of dig into, this is by each individual family. Each of these bars makes up how those birds were named. So for example, if you see an all orange bar, that means that all of the species in that family are named after physical traits. If you see a big purple bar, that means that all of the species, or that proportion of species in that family, are named after both sexes physical trait. And if we looked at which family of birds had the most species named after both sexes physical trait, and I think I had a cutoff of 10 species here. There had to be at least 10 species in the family to qualify. The answer for that is Buconidae, or puff birds, had the highest proportion of both sexes physical traits, so that's 92% of the birds. Mannequins, or Pipridae, had the highest proportion of being named after male physical traits. So 64% of species were named after physical traits only found in males, likely because many of the females are kind of overall green and look the same. Platysteridae, or the wattle-eyes and batistas, had the highest proportion of being named after female physical traits. So physical traits that are found in females, but not males. That's 12.5% of the species in that family. Megapodes had the highest proportion of species being named after geographic location, at 62% of the species. Musophagidae, or my favorites, Turacos, had the highest proportion that were named after people, so eponyms. And we finally did one last way we broke down the data, which was frequencies by geography. And so if we looked at the breeding range for each species, do we see trends depending on what part of the world they're from? So ocean species tend to be named after natural history, really all that's geographic location. If you think about many of the ocean-dwelling species, they don't have a lot of very distinct physical traits, so maybe that's not so surprising. Interestingly, Middle America and South America, and these designations are all taken from the IOC, the International Ornithological Bird Checklist of the World. The Middle America and South America had the most species named after physical traits and the fewest species named after people. Kind of Eurasia and North America had very similar proportions, as well as Africa So there's some really interesting, again, there's a lot of really interesting trends to dig in here. And one of the reasons we did this was because there's a lot we can then do with the data. We're excited to extend it to other languages. We're excited to think about, how dichromatic or how different males or females, does that change if they're more likely to be named after males or females? But it was a really fun exercise. It was a chance to get to look at birds that I'd never looked at before. And there are just a few kind of interesting stories that stand out. There are a lot of cool name stories. I will say one of the things that really stood out was how many ways there are to call a bird black or dark, other than the word black, which was, again, our most common term. There's cinereus, dusky, fuscus, plumbeus, saturnine, Stygian. So there are many, many ways to say that a bird is black. Another one of my favorites is the barnacle goose. So how did the barnacle goose get its name? Well, it's a myth that dates back to the 12th century, that says that they didn't know where these birds went to breed. So they decided that they emerged from barnacles under the ocean. Gerald of Wales claimed to have seen this species hanging down from pieces of timber and said that they had emerged from shells. So that's why barnacle goose is called barnacle goose, because they emerged from barnacles. Finally, my very favorite term was the Isabelline shrike, Isabelline wheat ear. There are a couple other Isabelline words, but you may not know this, but Isabelline is named after Isabel I of Castile. There are a couple of different myths here, but they think that this is what it is. And it's actually not named after her. It's named after the color Isabelline, which is kind of that dirty brown yellow color. And kind of the story is that Isabelline said that she would not, in fact, change her undergarments until the Moors had vacated Spain. So I think it took about 8 months. Now, I don't know if that's true or not, but when you look at an Isabelline shrike or Isabelline wheat ear, you might think, that Isabelline color. What an interesting story. So finally, I would just want to thank all of our participants who helped us categorize all these bird names. Again, it was a really, really fun exercise and fun to interact with lots of different people. So lots of fun bird happenings in the ornithology collection. We are currently hiring a collections manager. The search is open for a little bit longer. If you know anybody who is really passionate about bird collections, tell them to apply ASAP. And I have a lot more bird research stories to tell you, so you'll have to have me back for another talk at some point in the future. But for now, I'm happy to take a few questions.
Absolutely. We'd love to have you back sooner than 3 years from now.
Yes, hopefully sooner than 3 years.
Great. Well, thank you very much. And if you have a question, please pop it into Q&A. We have several questions already ready to go. And for the people on their live streaming on YouTube, we're going to go ahead and read. They can't see anything or can't see questions. They can only see you. And so we're going to go ahead and read those if you don't mind.
Sure.
First of all, our first question is from Yvonne, and she asks about those
the spectrophotometer.
Thank you very much. Are isolated feathers used, or does the whole body of the bird actually go into this?
That's a great question, Yvonne. So we actually tested both. So the whole body goes into the spectrophotometer. We tested individual feathers and they produced very different results. And so really you can't just look at one feather. You have to look at how they're layered on top of each other. It turns out it's really important. So we're hoping to quantify that more specifically. But yeah, it was the whole bird body.
Oh, interesting. She also asks, are down feathers able to control position by, I can't even say it, phylloerection? Phylloerection, is that correct?
Phylloerection, yeah. So that's like how birds are making their feathers go up and down.
Yeah.
That's a great question. Yeah, so our down feathers, so kind of that behavioral regulation of feathers and how that's related to thermoregulation isn't something that we studied here because museum specimens can't really move their feathers up and down. They're just positioned one way.
Right.
But that would be something to look for in the future. And so most of the feathers, the body feathers, they're half of it is that downy part and half of it is the colorful part. So they can control the position of those. And how puffed up are they versus how kind of more streamlined are they?
Right. Interesting. Interesting. Yvonne also asks, she's definitely monopolizing. Would you hypothesize that the feather-related thermoregulation strategies of male and female scarlet tanagers would be the same? Because obviously, because of feather coloring.
Yeah, so that's a great question, and that's like an active thing that I'm really excited to look at. So TBD, we didn't really use any species that had different colors, but I do expect they might have different thermoregulatory strategies. And yeah, I don't know, but good question. I would expect they might.
Fantastic. Interesting. Interesting. An anonymous birder lab member asks, do indigenous peoples' names of birds correlate mostly with the behavior or physical attribute? That's interesting.
Yeah, that is a great question. If we're talking about the local language names here, we don't really know because we don't know the translations of what those names are. So, yeah, I would love to know the answer to that question, but for the purposes of this first project, we don't know. And yeah, unknown. But that's, we are hoping to look at this in other languages, and indigenous languages could be one of those axes. So yeah, that's a great question.
TBD. So I take it a lot of this was spurred on by the AOS's project to get rid of eponyms.
Possibly, yeah, I don't want to get into that particular, should we do it or shouldn't we do it
Oh,
right
no,
now?
no, no.
That was what brought up this whole question was like, if we are changing bird names, what should we think about, potentially while we change them? Yeah,
Right. And how were they named in the past?
exactly.
Yvonne also asks, it would be fun to further it would be fun to further take the new common name and turn it into a new scientific name.
Yeah, thanks, Yvonne. That's a good suggestion, but the scientific name especially, we want that to remain stable just for the purposes of, having, linking bird names through time so that people could understand like what they're studying and things like that. And so we don't necessarily want to change the scientific name just because we changed the common name. But yeah, there are actually many bird names scientifically are not related to the common names at all. That's one thing that we found for sure.
But some definitely are related.
Some definitely are, but yeah, that would be another whole project. How many are related? I don't know.
And related by eponyms,
Yeah.
Anyways.
Yeah.
Okay. Yvonne also asks, would you like to suggest the Helm Dictionary of Scientific Bird Names by James Jobling, just for fun?
Yeah, that's a great point.
That's her suggestion.
Thank you.
Oops. And Sarah asks, can you give some examples of the birds that were hard to put into a category?
Yeah, there was, I would say, varied ended up being like a very difficult category because are we talking about varied in terms of, behavior, or are we talking about varied in terms of like if they have different types of features, is that really a physical trait or not? So we did have to make some decisions about those. Other sorts of things that were difficult were places that are kind of places, but they're also habitats. So like Chaco, for example, was tricky. And so that's why we ended up saying, if you can put a pin on it in the map, then we call that a geographic location. But if it's really a habitat, then and it exists in different places, then we called that natural history. So those were kind of like some of the trickiest ones.
Yeah, interesting, interesting. Carol asks, what is your personal take on how we should name birds in the future?
Yeah, great question, Carol. I think our results point to kind of underutilized categories of bird names like behavior. And so, maybe thinking about like song-related names might be a fun thing to think about. And yeah, or yeah, I don't know. I do think communicating something about birds is important, and behavioral would be really fun. So yeah, we'll see. I mean, there are a lot of different things, but that's kind of one of my takeaways. And thinking about what do we really want bird names to convey? So, if we
Yeah,
choose a really obscure color, is that a useful thing for people in terms of like what they're trying to learn about the bird?
true, true.
Yeah.
Anonymous birder also asks, my daughter asks, what your favorite bird is? What is your favorite bird?
My favorite birds are turacos. I love them. They're the only truly green birds, meaning they have a green pigment, instead of being a combination of blue feather structure and yellow pigment. So, yeah, those are my favorite birds, I have to say.
And where do you find turacos?
Turacos live in Africa. Although we did have a dead one show up in Beverly Hills my first year as curator here. Kimball will remember this, but I remember getting an email, dear Dr. Shultz, I found a resplendent quetzal in Beverly Hills. And it turns out it was not a resplendent quetzal. It was a white cheek Turaco, but we were really excited to get it for the collection.
It was even better. That's great. And I apologize to Surya because I'm obviously mispronouncing a name.
No, that's correct. Surya. He's
Surya.
going to be 11 on Thursday.
All right, Surya. Surya asks, give an example of a bird that was named for a trait only in females.
Ooh, good question, Surya. Putting me on the spot here to have to remember one of those 20 bird names that we had.
Well, I thought for sure it would be on one of our North American phalaropes but maybe not.
I believe redneck phalarope is one of those. I would have to check our database, but that the badises or the wattle eyes, that I remember that maroon throat was one of the ones. It was like maroon throated badis or something like that. But I will say, our database will be open to everybody. It's currently up on my GitHub in a very not user-friendly CSV file form. So we're going to work on a website for that. That's much more user-friendly, so you could explore all of these different names and kind of what their origin was in the future.
Fantastic. And believe it or not, those are all the questions. Thank you very much for a wonderful program tonight. Everyone, I think, really enjoyed it a lot. And for you people that are upset with me because I didn't put a question or riddle into the Q&A, it's into the chat, is because I was too fascinated with the webinar. I couldn't tear myself away. So you'll have to wait till next time. Alison, thank you very, very much. That was really wonderful.
Thank you. Very, very informative. Thank you for your great presentation.
Yeah, thank you for listening. It was really fun to share it with you. And I'll definitely, we'll have a lot more to talk about. So you'll have to have me again in the future at some point soon.
Okay,
Excellent.
we'll take you up on that.
Okay, great.
Excellent. Thank you all very much. And for everyone, don't forget, next month, we're going to be talking about swine hose, whatever. Well, we're going to talk about the white eyes. But the AOS will be changing all of that soon enough, so it doesn't matter. So, well, maybe not Swinhouse. But anyways, so we'll see you all next month. In the meantime, good night, everyone. I hope you have a good evening and get out birding. And take care. Bye-bye.
Thanks, everybody.
Great talk. Thank you. It was really nice and I learned a lot. Thank you.
My pleasure, Lily. Thanks for listening.
Thanks, Lily.
Welcome.
Good night.
Have a
Good night, everyone.